Hello and welcome to this lecture on adeno-associated virus and its use for gene therapies. We're going to talk about design and manufacturing of AAV gene therapy vectors. My name is Jørgen Magnus. I am head of the biochemical engineering at the University of Aachen in Germany. And this lecture is part of our lecture series on cell and gene therapy offered to the students in Aachen. So we will start by looking at gene therapies in general and give a general overview and classification of genetic diseases, because gene therapies are typically, but not exclusively, used to treat genetic diseases, but most of the therapies on the market today are used to treat genetic diseases. So we will give a short introduction to them. Then we will move on to look at AAV biology, look at how we can optimize the capsids and the genetic sequence, etc., for the AAVs using recombinant DNA technology. And then moving on to chapter 4, we'll look at different production systems on the cellular level, so the different expression systems that can produce AAV viral vectors. Then look at the production in large scales and the bioprocesses that are used in production facilities to produce AAVs. And finally, a quick look at the gene therapy LUXTURNA, which is a gene therapy that was launched in 2017. And it was the first, at least from the ones that are on the market today, this was the first one that was launched. So let me start with an introduction to gene therapy and genetic diseases in general. So a genetic disease can be caused by a monogenic disruption. In that case, it will be only one single gene that is affected. And if it's a recessive monogenic disease, you would have to have both alleles being affected by the wrong sequence of the DNA to have the phenotype, to have the disease. If it's a dominant disease, one of them will give the phenotype. But the monogenic diseases are maybe the easiest ones because it's a very clear biological function. So it's a very clear understanding on the cause of this disease. There is a gene sequence that is deviating from the natural DNA sequence and then producing a dysfunctional protein. And this protein is then lacking in the body of the patient. And what you do then with the gene therapy is that you add the correct DNA sequence and thereby allow the patients and the cells in the patient to produce the correct protein as well. And then you have replaced, in principle, the dysfunctional protein. And thereby, potentially, you have cured a genetic disease, which is something that seemed almost impossible only some years ago, because how can you actually cure genetic diseases that are present in all of the cells in the whole body? But as it turns out, and there are several examples now and several success stories about this, you don't necessarily have to correct the genes in all the cells in the body. It's only important that you correct them in those that are important for the function of that specific protein in the body. So we will look at this later in this lecture, how some of the very severe genetic diseases has been actually treated very successfully with gene therapies. But at the moment, in fact, all the therapies on the market are targeting monogenic diseases. The polygenic diseases are more complex, and they're actually more common than monogenic diseases, but they're also more difficult to target with the gene therapy, simply because it's not 100% understood always how these different genes affects the phenotype and the disease and how it leads to it, and what the cause of it is hasn't been maybe on the biological level understood to that degree that it can be targeted very specifically. And also when it's several genes, it becomes more complex. So this is maybe one of the remaining challenges that we can work to try to solve. And then there can also be, in some cases, you have large parts of the chromosome is missing, which is sometimes referred to as chromosomal aberration. So moving on to look a little bit more in detail on monogenic diseases and how they are inherited. Of course, monogenic diseases doesn't always have to be inherited. It can also be a de novo mutation, meaning that it happened just in the body of the patient, but that he didn't inherit it from his parents. But it's often inherited diseases, and in the case of a dominant inheritance that you see here on the left side, if you have an affected father, for instance, that has children with an unaffected mother, there will be 50% chance that he passes on his mutated gene, because he will have one mutated gene at least, and then he might pass this on either to a daughter or to a son. And the probability is then 50%. There's also the 50% probability that he get unaffected children without any defect mutated genes. In the case of the recessive inheritance, we will have, for instance, two parents that are both carriers of mutated genes. So the father has one normal gene and mutated gene, and himself is not affected by it because it's a recessive disease. The same for the mother, where also she has one mutated gene and one normal gene. And then in the case of their children, with a 25% probability, they can get a son or a daughter that had both genes on both chromosomes being affected by the mutation. On the other hand, there's also 25% chance that they get a child that is completely unaffected and is also not carrier of the defected gene. And in 50% of the cases, there will be a son or a daughter that carries either the mutated gene from the father or from the mother. And then looking a little bit more closely to a special case of monogenic diseases, which are the X-linked recessive monogenic diseases. These are related to genes that are present on the X chromosome, but not on the Y chromosome. And in that case, well, if we look at the left-hand side first of this slide, we see if there is an affected father that has a mutated gene on one X chromosome. So men only, of course, have one X chromosome and one Y chromosome. But then he will be affected because he has the mutated gene, and then there will be a 50% chance that he gets a child, but in that case it will be a daughter then, which is a carrier but is not affected by the phenotype, by the disease. If he gets a son, he has passed on his Y chromosome, and then the son has gotten a normal X chromosome from the mother, and therefore is not a carrier and also not affected by the disease. In the case of a mother that is a carrier of a mutated gene here, but is not affected herself because she has a second copy of that gene on her second X chromosome. Then if they get a son, that son will have 50% chance. So from their children, there will be a 25% chance that they can have then a child that has the X chromosome from the mother that is affected by the genetic disruption. On the other hand, their other children with a 50% probability will then be unaffected and also not carriers, but then they can also have a carrier daughter that is unaffected by the disease because she still has gotten the correct X chromosome from the father. So these are X-linked monogenic diseases and how they are inherited. And we will focus on in vivo gene therapies in this lectures. Before I move on to talk about that, I also want to mention that there are also ex vivo gene therapies. So the ex vivo gene therapies means that you will take cells out of the body of the patient, and then you will modify them genetically in vitro. So this happens here. And then you will send those cells back into the body of the patient, where they can have a new function and can potentially then treat a disease. So the most prominent example of this is the CAR T treatment, where you take T cells out of the patient, you then give them an antigen receptor that they didn't have before, and that they express on their surface, and then they can attack tumor cells more efficiently, for instance, to be used to battle leukemia. And in this case of the ex vivo gene therapy, that would also be a cell therapy because the product is then cells. We then distinguish between autologous and allogeneic cell therapies. Autologous is if the cells comes from the own patient, so he gets his own cells back again. And then if it's an allogeneic cell therapy, these are donor cells that can be given to a different person than where they came from originally. But in both cases, you would typically do the genetic modification by integrating these genes on the chromosomes of the cells. And there it's typical to use lentivirus to do that. But you can also use a gene editing strategy, for instance, using the CRISPR-Cas system, where you would provide a nuclease like the Cas9 protein together with the guide RNA, and also a plasmid that carries the sequence of the gene that you want to express. So in the CAR T example, it would be the antigen receptor. And then you will treat them in vitro and then send them back to the patient. But we will focus now on the in vivo gene therapies. And moving on to the next slide, there are different possibilities and maybe also other possibilities than I think I maybe have shown here, the three most common ones. The one that has been most successful until now is the so-called gene replacement that is used by AAV. And that is also the focus of this lecture. But just to mention gene editing as well, which is also an exciting opportunity. You can also do in vivo gene editing. In that case, you still need to provide your nuclease, but you would do it, for instance, by using an mRNA formulated in a lipid nanoparticle, and then you have to connect it with the guide RNA, and then you could use an AAV vector actually to provide the sequence of the gene that you would like to integrate into the cells. So what happens actually in the gene editing strategy is that the missing gene that you would like to replace is actually integrated on the chromosome and also at a specific site. That's why you need the guide RNA in the chromosome, and then it can be expressed from there. In the case of the gene replacement using AAV, it is not integrated on the chromosome, but the DNA of the viral vector is simply released into the nucleus, where it's present as a free DNA molecule, and it's not connected directly to the chromosomes. But as a free DNA molecule, it can still express the genes, and it can still produce the protein that is lacking in the body of the patient. And that is a strategy that has been proven in many examples to work well. We should also mention the antisense oligonucleotides here in the middle. This can affect the gene expression more on the transcriptional level, for instance, by affecting the mRNA molecules and reduce, for instance, the expression of certain genes that can have also a therapeutic effect. So moving on here to talking about the viral vectors. You can deliver them by using nanoparticles, for instance, lipid nanoparticles, same technology that was used in the COVID vaccine. But the viral vectors themselves and the virus, by evolution, has developed to be able to integrate into the cell and into the nucleus of the cell. And so that is part of its nature to actually do it, and therefore it goes very efficiently. And this is really the method that has proven to work well, and it will be the one that we will focus on here in the next in the next slides. So just a quick look at how the adeno-associated viruses are constructed. These are replication incompetent virus, which means that they cannot reproduce without the help of the so-called helper functions that can be provided by an adenovirus. And this is why they're called adeno-associated virus, because they were discovered next to adenovirus, which are much larger virus. And they can only replicate if they get these other functions, these other enzymes from the adenovirus. There are also other types of virus that carries these helper functions, but most typically it's the adenovirus that is associated here. So they are non-pathogenic. They have been classified, in Germany at least, as biosafety level 1. This goes for all the most common types, the most common serotypes AAV. There are about 13 different serotypes that has been isolated, but the most common ones in gene therapy, like the AAV2 or the AAV5 or the AAV6 or 8 or 9, have all been classified as biosafety level 2, which makes it, biosafety level 1, which makes it easier to work with them in research, and also easier to apply in a therapeutic environment. So that is good. And they are very, very small. They're depends a little bit on the serotype, but they can be between 22 and 28 nanometers big. They have a single-stranded DNA genome, and there's a low risk of chromosomal integration. This has been observed in mice. It has also been observed in cell-based assays that there can be chromosomal integration, but it's very, very seldom. It has, to my knowledge, not been observed in human beings. But there is a very, very small risk of chromosomal integrations, which is different, for instance, from lentivirus, and is a huge benefit in the risk assessment and the safety aspect of adeno-associated virus. Yeah, so looking at the timeline here, they were discovered in the 60s and further developed and further discovered in the 70s and the 80s. The first human application was in 1995. There was a little bit of a setback in 1999 when one patient actually died in a clinical trial. This was the very early days of AAV gene therapy, and the patient had a condition that was not known at the time when he received the injection of AAV virus, and therefore it sort of set back the whole development a little bit, because it became clear that there is also a significant risk associated with these treatments. But then in the last years, and the last, I would say, 8 or 10 years, there has been a very strong increase in research initiatives, in clinical trials. There has also been several market launches. We see here the first FDA approval of LUXTURNA in 2017, and after that there has been several further ones, which we will see on the next slide. And here we see the list of the currently approved gene therapies. We see in the recent years, 2023, 2024, there has been 4 new ones that came onto the market. They all target monogenic diseases. They all target recessive diseases. Some of them in the recent years target also these X-linked diseases that affect predominantly men. These are hemophilia A and B, and also Duchenne's muscular dystrophy, which is a very severe disease that also leads to an early death of the patient, same as the spinal muscular atrophy with Zolgensma in 2019. which was sort of the second success in gene therapy after LUXTURNA. And then the obvious challenge here for society, I would say, is the very high price of these treatments. As you can see, they sometimes go about 2 million and sometimes about 3 million per patient, which is a difficult cost for healthcare systems to carry. The fact that they do it just shows how important they are, and sometimes life-saving treatments. But if this should be offered to a larger portion of society, you would struggle with financing these high costs. And therefore, we should work to reduce the costs, but just understanding why they are so expensive. It's not, I mean, the manufacturing costs are at the moment very, very high, but still, and it also depends a little bit on the indication and also the type of viral vector that you're using. But I believe that also the manufacturing costs, or let's say the processing and the production of material for clinical development plays maybe an even higher role, because if you have a robust process and you have reliable and consistent quality of your product, and you can always produce at a low cost in a reliable way, that could significantly speed up the timeline of clinical development, because in fact most of the clinical holds are actually due to CMC topics, or so-called CMC is related to the production of and the quality of the material that's been delivered, and there's still a lot of work that has to be done to be able to establish really very good production processes, and this can be a leverage for also reducing the cost of these very important therapeutic options. So the other aspect here, of course, is that they're all used for rare diseases. Therefore, there are very few patients that have to pay for the development cost in a way, and maybe the development cost can be as high as $1 billion over 10 years. And in that case, you would also have a return on investment that the people who have invested in the pharmaceutical company would expect, and also the years after the launch that has to be repaid. So what you're repaying is actually much more than that 1 billion. And that makes it very difficult for pharmaceutical companies to calculate a positive business case for this, and it only works out when they demand these high prices. So it's a challenge, but it's partly also a technical challenge that we as researchers at university can try to do something about. So moving on to the AAV biology, and this will be just a short introduction. We have the AAV genome, which is limited to 4.7 kilobase pairs, and or kilobases. It's a single-stranded DNA molecule. So we have between the two ITR sequences, there's a maximum of 4.7 kilobases. In the wild type, this is the rep and the cap genes, and then this inverted terminal repeats that you see here on both sides. So you see them here and here. This needs to stay, but when you construct your recombinant AAV vector, you will remove the native sequences of rep and cap, and then this is where you insert your gene of interest. And there's also poly A chain here at the end of it. And then this vector genome is located inside the capsid. So it's a non-enveloped virus, so there's no lipid membrane around that again, but there's a capsid, a protein capsid where you have space enough for this DNA. But if you try to make it significantly bigger than this wild type size of 4.7, you will run into problems, and you will have lots of incomplete genomes in your capsid, and in general this is the guideline that not to exceed the 4.7 limit. So we have the 4 rep genes here that are important for replications and packaging, and then you have the viral capsid, or the protein particles in the capsid that is coded by the cap genes. And there are 3 of these viral particles. They have a ratio of 1 to 1 to 10, and they assemble into a capsid, and then the vector genome that is produced separately can then be packaged into this capsid. The surface of this capsid is important. It decides the tropism of the capsid, so the ability to incorporate into different cell types. So AAV vectors, they often end up in the liver, but you sometimes want to target maybe a muscle cell, or maybe you want to target a nerve cell or something, and then you would have to have specific properties of this capsid to be able to direct them more towards a specific cell type. And that's why the different serotypes is because they have slightly different tropisms, and you can choose the serotypes that is most suitable for your gene therapy and for your application. The virus then gets into the cell by attaching to a surface receptor. It's then incorporated into the cells. It sometimes happens that it's then just dissolved, and it's attacked by proteinases who dissolves the capsid, and then nothing more happens. But in some cases, the AAV vector managed to get integrated into the nucleus. So as you can see here, and then it can release its DNA molecule into the nucleus of the cell. And in that moment, it's a single-stranded DNA molecule, but then they can synthesize the second strand, and then you have a double-stranded DNA molecule. Or in some cases, if you have organized to have the complementary strain already delivered, maybe as part of the AAV vector, then you have a faster assembly of the double-stranded DNA molecule inside the nucleus of the cell. And then, as I mentioned before, it's typically present as a separate independent DNA molecule inside the cell, but in some cases, or it could, at least there is a potential very small risk that it could also integrate into the host genome of the chromosome, and that is in general something that you would not want to happen. But as mentioned, the risk is very low for that to happen. In the replication of the virus, it will then create its capsid from the virus particles. It will then start replicating itself from using the rep genes, but also with the help of the helper genes. So in principle, it needs 3 elements. So it needs the cap genes to produce the capsid, but it needs also the rep genes, and then it needs the virus genome. But in addition, it also needs helper functions that can be added by an adenovirus. And this is how the wild type is replicating, only in the presence of an adenovirus or another virus that provides the helper functions. So then what can happen is that not all the capsids are full with a virus genome. It can also happen that empty capsids form. In the wild type, it's almost 100% full capsids, but producing the recombinant, so the modified AAVs, you can often end up with a very low ratio of full capsids, which is obviously not something that you want. You would have as many full as you want to, but then you have to deal with this in your purification process and sort of get rid of your empty capsids to have a product with high quality that is mostly full capsids. Now we will look more into how to optimize the characteristics of the AAVs using recombinant DNA technology. starting with the capsid engineering. The capsid is very important in at least 3 aspects. One of them is the tissue tropism, because as I mentioned, you would like to attack certain cell types and certain tissues specifically. That can be optimized by changing the sequence of the proteins or the amino acids strictly, or in principle the DNA sequence of the cap genes, to create capsid that have different properties. Also, it's important to avoid the immune system, because when you inject virus into the body, in principle, your immune system would answer and defend itself against this virus. And therefore, to try to avoid the immune system of the patient would be also an important feature of the capsid. On the other hand, you also do immunosuppression during the treatments, and there can also be there can be different ways of doing this. One strategy is to cleave the neutralizing antibodies, or you can use corticosteroids for immunosuppression. But ideally, you can avoid strong immune response from the patients, and if it's too strong and if it's too high dosage, it could also be dangerous. And thirdly, and this is maybe a topic for research that is interesting and still ongoing, is to increase the packaging capacity of capsids. So that it could be potentially possible to use a larger genome size at 4.7 kilobases. So we have all these different technologies. You have directed evolution, which is based on random mutations and iterations, where you can use DNA shuffling or error-prone PCR, peptide display, and also other different types of molecular biology to generate different variants and then screen them. Rational design, if you have a clear idea of the sequence that you would like to target, you can also do targeted genetic mutations to the cap genes. Or maybe a new method is to, if you have a lot of data about the different effects that you have from different changes in the sequence, you can use machine learning to extract sort of the understanding of which modifications would lead to which features of the capsid. So these are so this is an active and very interesting field of research to develop new capsids and better capsids. One interesting aspect is if you have to target the brain, and if you're developing a gene therapy for the brain, you would have to typically introduce the AAVs directly into the brain, because it's very difficult for the AAVs to cross the blood-brain barrier. But recently, some capsid has been developed, some recombinant and modified capsid that is able to cross actually from the bloodstream into the brain. And by that, you would avoid this surgical procedure of injecting a needle into the brain of the patient, which is an obvious advantage and reduces also the risk or increases the safety for the whole therapy. And this has been achieved by different methods of directed evolution and other methods to develop new capsid that has these properties, and allowing for a systemic injection also to target the brain. So that's the capsid engineering. The genome engineering sometimes, one challenge, of course, is that with 4.7 kilobases as the maximum, there are certain proteins or certain genes that are simply too big to fit on an AAV vector genome. This goes, for instance, for the dystrophin, which is the protein that is lacking in Duchenne's muscular dystrophy. And therefore, what is currently being used is a microdystrophin, so a smaller version of this gene that still gives a functional protein, which is still not exactly the same protein as the normal protein, but it's something that still has an activity. This is, by the way, also the same in the case of hemophilia A, where they deleted the B domain from the factor VIII gene or the factor VIII protein, so that the gene for that would fit also on an AAV vector. But what you could also do is to simply split the gene in two and use two different AAV vectors. Then you would have to combine them later so that they yield the full length protein. But there are also different tricks on how to do this. And so that's something that could be considered for those special cases where your gene is too big for to fit in its full length on the AAV vector genome. Another interesting trick that has been used actually in Zolgensma, which is the gene therapy for spinal muscular atrophy, they included a self-complementary AAV. So they included the complementary region of the single-stranded AAVs that then combines inside the nucleus of the cell, and this means that the formation of the double-stranded DNA goes much faster than in the other case when you have to synthesize it from nucleotides in the cells of the patient, and therefore makes it more efficient and allows it maybe to dose lower and has significant advantages by doing this. But obviously this only works if you have a very small gene, because then you would have to fit both the complementary strain as well as the normal sequence on one AAV vector. So that works if your sequence of your gene is less than half of 4.7 kilobases. And then the vector genome itself, you need, of course, the transgene itself. So this is the most important part, the gene of interest, sometimes referred to as payload or gene of interest. You need a promoter to be able to express it. This promoter could be specific for a specific cell type, and that can be interesting if you want to have expression in certain cells, but in other cells you would like to avoid expression. You could carefully choose your promoter to fit that purpose. There can be an enhancer as well that is included that can enhance the strength of the expression, or also regulate its transcription. The poly-A tail is needed also for transcription, and in certain cases you could include a sequence here that is helpful for the localization or for the movement inside of the cells. If, for instance, you would like to move the protein back into the nucleus of the cell, if there is some kind of regular functions that you're targeting, you could include a localization signal that helps with moving that protein over the membrane. Or, as is, for instance, the case for the hemophilia A treatment, you would like it to be moved out of the cells. You could also include a sequence or a small peptide sequence that helps the protein to move across the membrane out of the cells and into the bloodstream, where the factor VIII protein that is supplied in the hemophilia A treatment is needed. So these are the elements, and as you can see, they're both located between these ITR sequences that are here on the both sides of the genome, and including the ITR sequences, you have a maximum of 5 kilobase pairs. I think the ITR sequences themselves are 154 bases, so the 4.7 in between them plus the ITR sequences gives you the 5 kilobases. That is the maximum of the vector genome. So now we're going to look at AAV production systems on the cellular level, and we will look at these three different production systems: the stable production cell line here, the ones that is based on the baculovirus, can also be based on herpes virus. This is essentially the co-infection system, which we will see in a minute. And then we have the triple transfection system, or the helper-free transfection, which we will also look at in the third chapter here. Essentially, and maybe as an introduction, there are three different factors that you need to be able to express AAVs. And this is, on one hand, the rep and cap genes. It's, of course, the viral vector itself, the genome of the vector, so containing the gene of interest. And then it's the helper functions that are needed for replication and that are in the wild type case provided by the adenovirus. So one way to produce AAVs is to create a stable production cell line. And this can be done by integrating the rep cap genes and the gene of interest on your genome of the cells. So you can take, let's say you take, for instance, a HeLa cell, which is a human cell line, and then you integrate the rep and cap genes as well as the gene of interest on the chromosomes of that production cell line. Then the only thing you would have to do is to add the adenovirus, because then with adenovirus comes the helper function, and then you have those three elements that I said, the rep cap genes, the gene of interest, and the helper functions. And then you will have production of AAV capsids. Another way to do this, maybe not as established, is to use capsid that aren't integrated on the chromosome but are still retained in the cells, maybe by using an antibiotic market or something like that. But I think, at least to my knowledge, the stable production cell systems typically uses genomic integration. So that's one way to do it. Another way to do it is the system based on baculovirus. And baculovirus can infect insect cells, and you would use the SF9 insect cell line to do this. In that case, the baculovirus actually has the helper functions itself on its genome. So you only need two other elements, which is the gene of interest here and the rep cap genes. So you would create two different strains of baculovirus, one that carries the gene of interest, one that carries the rep cap genes, and then you would co-infect your insect cells. And when the insect cells are infected at the same time with both of your baculovirus strains, then they will produce AAV capsids. And the same way for herpes virus, in that case you can use a mammalian cell, so you can use a Vero cell line, for instance, is typical, which is a cell line from a monkey but not from a human being, but still much more similar to human being than an insect cells. But herpes virus hasn't been used for different reasons as much as the baculovirus system. So there's always a safety aspects involved, and the baculovirus only attacks the insect cells, and that makes it a little bit safer for human beings in the production process. There are some disadvantages and advantages. I think most importantly, the post-translational modifications of baculovirus or insect cells could be different than in human cells. So maybe the glycosylation pattern is different on the surface of the capsids, which gives you then a capsid that is slightly different from what you would otherwise have when they're produced in human cells. But it has been a technology that has been used, and it has been used partly because insect cells are easy to cultivate in suspension cultures, whereas human cells, at least in the past, have often been cultivated in adherent cell culture. And then the most common, and this is certainly more than half, or at least I would say 70% of all processes depends on this triple transfection process. Here you have 3 different plasmids that carries the 3 different functions. These are the gene of interest, the rep cap, and the helper functions on 3 different separate plasmids. They are mixed with a transfection agent like polyethylenimine, and then they can be transfected into, for instance, hex cells, and typically it's the human embryonic kidney cells that are used for this. And then once you have all 3 capsids inside the hex cell will be able to produce AAV capsid. This is the most flexible way to do it. As you can see, there's no genetic modifications needed in the hex cell line. And you can also change very easily if you want to produce several different types of gene therapy. So you can change the plasmid with gene of interest, for instance, to produce a different type of cell types as gene therapy. Or you can change the rep cap genes to have a different capsid, a different serotypes of viral vector, and so it gives you a lot of flexibility. It also gives you very high costs of plasmids because they all have to be supplied as GMP quality, and when you produce them yourself, you would have all the effort of producing, and you can produce them in E. coli, but then you would have a GMP process in E. coli, or you can buy them, but then they're expensive, of course, and you need a lot of it. So the cost at large scale becomes very, very significant with this. We're moving on to the next chapter, which is chapter 5. And looking at how these are produced at large scale. And first, I would like to show an overview of what I have maybe already mentioned. So there's a lot of information on this slide, but if you look at the left-hand side here, you see the process steps where you have the manufacturing of the cell banks, the manufacturing of the infection solution or the transfection solution, depending on your production system, then the cell expansion and the fermentation, and the production of virus, and then the purification, which can be also very complex. Here it's just one line, but there's of course several different unit operations that is needed to purify the product at the end of it. And looking at the stable production cell line, and just to give you an overview of the differences here, as I mentioned, the HeLa cell line is typical in when you make production cell systems. Here you would have to do all the cloning work, and you would have to establish the cell bank, and you have to establish a master cell bank that is also GMP qualified. That takes a lot of effort. And then you also need to produce your adenovirus, which again is also another bank, or it's another master cell bank or a master virus bank in that case, that you also need to add in your infection process here. So that becomes more complicated and has several more elements. And then when you come to the main fermentation down here, you have your main culture with HeLa cells, and then you grow them for a certain number of days, and then you add the adenovirus, and then you have a production phase, and then you harvest and you do the purification. In the baculovirus case, or the herpes virus case, you do the co-infection, as I explained, and you add the 2 different types of virus, and then you do the main culture and infect your main culture the same way that you infected your HeLa cells with adenovirus. In both of these cases, virus safety is a topic, especially when we look at the stable production cell system with the HeLa cells and adenovirus. At the end of it, you have produced a lot of adenovirus, and you need to be very careful to separate them out from your product at the end of it. Adenovirus are much bigger than AAVs, and they're also more sensitive to heat. So that's something that you can use, for instance, having a heat inactivation step where you can inactivate your adenovirus but still keep your AAVs intact. Just have to be careful how to set the temperature. But it's an additional safety aspect that we don't have in the third system here, which is this triple transfection, where you simply need those 3 plasmids, but then of course you need to produce them, and you typically produce them in E. coli fermentation first, and then you add in the transfection process, you add them to the bioreactor. So here it's typical. You might have a growth phase in your main bioreactor of 4 days, and maybe have a production phase of 3 days. And after that, you have produced so much virus that the cells in your bioreactor is significantly impacted by this, and it doesn't make, there's no advantage in continuing for a longer period of time. And then you do the harvest. So that's how you do it in the case of the triple transfection. And in that case, you still have to separate out your plasmids that you will inevitably have in your final product as well. But since they contain also an antibiotic resistance, it's important to separate them out and to get them down to a very, very low level before you release the product to be used in a clinical trial or in a production in a therapy. So this is an overview of the 3 different expression systems and their processes. We will focus now on the production, but we won't say so much on the processes here at the beginning of it, where you prepare the cell banks and the manufacturing of the infection or the transfection solution, but focus here on the main production and the purification. Adherent cell culture is a technology that was used in the past before suspension cell culture was developed. And when you have adherent production processes, you need a lot of surface for the cells to grow on. And if you need a lot of cells to produce a lot of viral vectors, then you would also need a lot of surface. And this leads to, so you use these plates, and they are 632 square centimeters in surface, and then you can pile them on top of each other. So here you have 40 plates on top of each other. So this is not scale up, it's scale out, because you're simply increasing the number of plates. And then you can take those 40 plates and you can add them, 4 of them on a cart, and then you can have 4 of these carts into an incubator. And now you're having in this incubator 40 plates times 4, which is 160, and then times 4 again. So you're at 640 plates in your incubator that needs to be handled manually, and that has poor quality control and no monitoring of dissolved oxygen or pH or other things that you would like to control in your culture, and therefore isn't a very good production technology. But before suspension culture was developed, this was the only production technology. So moving on to what most people is using today, this is the suspension culture. And here you can use a classical bioreactor, like a stirred tank reactor. You can start in shake flasks if you want, but then move it into bioreactors that get increasingly big. Here is a picture of a bioreactor in 2,000 liter scale, and that is really the production scale of AAV viral vectors, where you would simply scale it up in several different steps into the 2,000 liter bioreactor. Looking a little bit more closely at this process, we would start with a cell bank and a vial from a cell bank, and then we would expand it into shake flasks. You can use a BioWave 20 liters or 2 times 10 liters, for instance, and then go into a 200 liter and a 2,000 liter. So in this example, you scale it up by a factor of 10 for each of the steps. You can also use a factor of 5, and you can also use different tricks on how to do this. You can intensify the process using, for instance, perfusion in your N-1 stage and create more cells. And then you can start with higher cell density in your production bioreactor and potentially increase your output significantly. So this is just a standard process. What we see here, it's a typical process. We also see up here that the transfection solution, and this is in the case of the triple transfection, has a significant volume, 10% of the production volume. And there you have the 3 plasmids that are mixed with the transfection agent, and then they need to be carefully added to the main bioreactor. And there it's very, very important that you have the right mixing times and the right mixing characteristics, the right shear forces in that mixing, and that you have a good mixing into the bioreactor, which isn't that simple at such a large scale. And also you have to remember that that cells have to be handled very carefully, so you can't stir at very high stirring rates, for instance. So that is one of the critical process parameters of this whole process is actually the preparation of the transfection cocktail here in a separate tank, which at production scale is a rather large tank at 200 liters. Moving on to downstream processing and the harvest. So once you had so let's say in an example, 4-day growth phase and 3-day of production phase. So if you run your bioreactor for 4 days and then you add your triple transfection, your 3 plasmids to the bioreactor to start the production phase, then after 7 days you can do your harvest. And then it actually depends a little bit on the serotype that you have. So some serotypes tend to remain inside the cells and some tend to escape the cells outside. But in some cases, you might have half your product inside the cells, in which case it makes sense to do a cell lysis. So add a detergent or something else that breaks the cells and releases the AAV vectors inside the cells into the medium. But if you do that, you will also release everything else that's inside the cells, for instance, the host cell DNA. And so we will have lots of DNA swimming around in the fermentation broth. That makes it a very slimy and viscous liquid. And so you can add, for instance, a benzonase that dissolves this. This is an enzyme that cleaves the host cell DNA, and thereby you can reduce the viscosity of the broth, or you can do a flocculation step. But in any case, you have to do something before you go into your depth filtration, because in your depth filtration, you will very quickly block your filters if you just do an untreated fermentation broth directly onto the filters with all the host cell DNA. But then the most interesting separation steps here is the chromatography steps here, where you first do capture chromatography. So in that case, you will retain the AAV particles and essentially let everything else flow through. And by that, you can actually get rid of 99% of your impurities already in one step. Then in the second step, you would separate the full and the empty capsids. And traditionally, this is done by centrifugation. So by ultracentrifugation at very high G forces, you separate your empty and your full capsid based on the differences in mass, actually. So the full are slightly heavier than the empty because they contain a DNA molecule. And you can use this to separate them by gravity, by gravitational forces. And finally, you would do a chromatography polishing step. And this is just simply to get all the impurities down to the very, very small level that is required for product release. And that is sometimes based on ion exchange chromatography. There's also a lot of effort to develop ion exchange chromatography for separating full and empty capsids, and some companies are moving in that direction already, but the classical and the most established way of doing it is the ultracentrifuge. And after that, you exchange the buffer to get the buffer that is needed in the final formulation, and then you can fill your vials and send it to the hospital. So the details here of the capture chromatography is that you have an antibody fragment that is able to bind the capsid of the AAV and then retain it in a chromatographic step. You can load at a neutral pH, so around 7, and then you can elute it at the at the lower pH, for instance 3.5 or something like that. And this is how you do a capture chromatography, by a bind and elute function. The separation of empty and full is done at very, very high G forces. So you centrifuge. You can also do it at slightly less than 350,000 Gs, but you can go up to 350,000 Gs to separate empty and full. And then you will have a fraction of full AAV particles at a certain height in your sample tube. And then you have to extract them manually. That is a very manual process, and it is a bit of a it's a more challenging step in the production because it's difficult to scale it up. So in a large scale, you will often have many different ultracentrifuges that run at the same time, and they take time, and it's also a delicate procedure that requires a lot of training for the operators who are doing it. So it is sort of a bottleneck in principle in the production process, and it's a reason why everyone would like to have a better method, like a chromatography separation of empty and full rather than the ultracentrifugation. And the solution to that could be an ion exchange chromatography, because the empty and the full capsid will have slightly different charge on their surface, because the DNA molecule itself is slightly negatively charged. So the full capsid would have a different surface charge than the empty capsids, and that can be used in an ion exchange chromatography. But since the difference is so small, it's a challenge to develop this. So it's still under development. But coming to the polishing chromatography, that's also typically ion exchange, and here you will have the classical way of doing it, that you can bind. You could in principle also use a bind and elute strategy, where you bind the AAV capsid to a surface of the opposite charge, and everything else can flow through. Potentially you could also have a flow through chromatography, where you just bind the impurities and you let the AAV capsid flow through. So there are different ways of doing this. So these are the most important steps in the downstream processing of the AAVs. Now we're moving on to analytics. Analytics is a very important part of production. You always have to make sure that your critical quality attributes are being met within their predefined limits, and there are many different characteristics that you need to measure before you can release it, and there are many different methods to do this. Some of them still in development. This is of course a new field, so there's always new and better methods that are being developed, and we should mention only a couple of the most important one here. So I mentioned the difference between full capsid, partially filled capsid, and empty capsids is very important, and you should probably go to at least 70% of full capsid, or better 80%, or better than 80% of full capsid, with the partially filled capsid being very low, ideally less than 1%, and the purified, the empty capsid then at least, I would say, less than 30 or less than 20%. Aggregates is a problem with some serotypes, and not such a big problem with other serotypes. But in general, you also need to have very low rate of aggregation. So if it's the small aggregates, you might be okay with 2%, but the large one should be at a very limited to less than 1 parts per million. Other interesting assays, the infectious titer is measured in cell-based assays in production. In research, you could, in principle, also use in vivo investigations in mice, but I think in the production environment, you would use cell-based assays. Then you can find your infectious units per milliliter, which is a very critical measure, of course, and important in setting the dose. The genome titer, maybe the most important measurement, is typically measured by some kind of PCR. It can be qPCR or other types of PCR to measure the genome titer. And then you can determine how many vector genomes you have per milliliter, which is essentially the specification of your dosage. And then the capsid titer can also be measured by HPLC, for instance, and there you can also see the difference between the empty and full, and also get an impression of the empty and full ratio. So rounding off this lecture with an example of a gene therapy, and we chose LUXTURNA to just have a look at LUXTURNA. LUXTURNA, as I mentioned, is the first gene therapy launched in 2017. It treats an eye disease called Leber's congenital amaurosis. This is a genetic, it's a monogenetic disease that causes blindness, and sometimes it also causes blindness at a relatively early stage in life, so it's a severe disease. It is caused by a disruption to the gene RPE65, which is involved in the regeneration of vitamin A in the eye when from the trans form to the cis form. So if this protein is missing, or if its function is missing, if you have a dysfunctional RPE65 protein, then you won't be able to regenerate vitamin A, and then you won't be able to sense the light anymore because you don't have the cis form ready to detect the light in your retina. And LUXTURNA is actually injected locally, so it's an injection into the eye and into the neural retina of the eye on the back side of the eye, where these AAV vectors act locally, and they integrate locally into the cells there, express their protein locally. And as I mentioned, this is a potential cure, even if you have only applied it to a very local area, which is the eyes. Whereas all the rest of your body, all the other cells in the rest of your body are still having the defect protein, but it doesn't make a difference because that protein is only needed in your eyes. So that is the potential of gene therapies, that you can treat them also locally. Here we see the 10-year development period of LUXTURNA, which is rather typical. You would hope to do it faster than 10 years, maybe in 7 years. I think most optimistically, you could potentially carry out a clinical development in 5 years, but that would be super fast. Here it was submitted as an investigational new drug in 2007 and then got approval in 2017, 10 years later. And interestingly, they actually use adherent cell culture because they use roller bottles to produce it. And that seems to some people like an ancient technology, and that roller bottles isn't really state of the art anymore. But in this case, since you need so little material for treating this eye disease, because you're only applying it to the eyes, so a very small volume is needed, and only here you see the dosage is 1.5 times 10 to the power 11. That's all you need per eye. It's okay to produce it in roller bottles because you don't really need that much material. In other gene therapies, where you go up to, for instance, 1.1 times 10 to the power 14 virus genomes per kilogram of patient, and maybe you have a grown-up patient, you would need really orders of magnitude of more material to treat that patient. And in that case, if you try to do it by roller bottles, you would also need orders of magnitudes of and several orders of magnitudes the amounts of roller bottles, which just wouldn't work, because you would really have an enormous space of roller bottles. And that's where it's important to use the suspension culture to produce the drugs. There's also different expedited programs that you can get from the FDA when you develop these therapies. They're all used for very severe and rare diseases, and so they often get this breakthrough designation or this RMAT designation or the fast track designation that speeds up the process of developing the or the whole regulatory process and the clinical development a little bit. But you still, of course, need to show safety and efficacy, which is almost important. Finally, just an interesting insight into how this was developed and how they set the endpoints of the clinical development. You could, of course, ask patients to read letters that becomes increasingly smaller, just like you do at the opticians, and just to check how small letters you can read and what the difference is after the treatment and before the treatment. But what they also did here was to ask the patients to walk through an obstacle course at different light intensities, because it's really the ability to see at poor light conditions that is affected here, and it's in particularly at low light intensities that it's difficult to see with this disease, and therefore it made sense to check how well you can see at in darker conditions. So there was a significant improvement in the light sensitivity, and also interestingly, since this was a very local injection at a very low dosage, there was no adverse effects relating to the virus itself. Of course, the surgery and the injection of the needle into the eye is in itself maybe a risk, but it's also an established procedure. But in this case, since the dosage was low, the virus themselves didn't pose so much of a risk to the patient. That is different in systemic injections, where you can have very severe responses by the immune systems that can lead to a very long time where there is fever and headaches and nausea and other kind of symptoms as a reaction to this immune system. But in this case, with the local injection, this was not the case. So that was just a brief look into LUXTURNA and one of the successful therapies. And I hope that in general we could provide you with a first insight into AAV gene therapy and the design and the production of it, and hopefully a motivation for looking more deeply into this. It's an enormous potential to cure very, very severe diseases, and there's a lot more that is going to come in the next years. There's a lot of clinical developments going on, so it's going to be interesting to see how this can move onto the market, and hopefully it will also be possible to reduce the cost of these gene therapies so that it's possible to get them covered by the different health care systems. And ideally, they're also made available to countries outside of the rich world, which is another aspect of this, that with such high cost, it's only very few countries that can actually allow themselves to offer these therapies. So with that, I would like to close the lectures, and thank you very much for the attention.